Animal engineering is the altering of DNA of animals for a particular trait. It can be inserting human DNA into animal DNA, cloning DNA, or selective breeding. Xenografting is the use of human DNA to supplement in animals or vice versa. This has become possible over the years with new technology. We use animal tissue and put it into humans, or human tissue into animals for testing or repairing damage. Cloning is the copying of a full genome. The first cloned animal was Dolly the sheep. Scientists have been developing cloning, but still run into mutations in the new sequences. This results in high failure rates, problems during later development, abnormal gene expression, and telomeric differences. Telomeric refers to how short the chromosomes are after being copied into the cells. The ends get progressively shorter throughout the organism's life, which has an effect with dying. Selective breeding is where humans match up animals according to favorable characteristics. This can be seen with domestic animals, such as dogs, and farm animals, such as cows and horses. The FDA has started to regulate genetically engineering animals. People who are genetically engineering animals must show that their products are safe for the health of the animals and for food consumption. For research and development, we have been using cells to test genetic engineering. Cells such as stem cells or HeLa cells are used. Stem cells are cells that have the potential to develop into many different cell types in the body during early life and growth. They are unspecialized and can be renewed through cell division. They can be induced to become certain types of cells depending on outside factors. They repair damaged tissue or organs. HeLa cells are cancer cells that never stop dividing. They originated from a woman named Henrietta Lacks, who was alive in the 50s. A sample was taken from her tissue, and they have not stopped growing since. Many scientists use these for medical research because they are easy to replenish. They need Luria broth, or food, and they keep growing. There have been ethical concerns with her cells because the doctors did not have permission from the family or Henrietta to use her cells for research. With the genetic field growing, there has been a high demand in organ and tissue transplants. Many people have used tissue transplants in cancer patients, car accident victims, etc. The market for organ and tissue transplants have increased over the past few years. The one thing we need to watch out for in the future is the legal rights. This has been a huge deal in the genetics industry. Many people have been concerned about the rights to these tissues and who gets ownership once they have been removed from the body. This was the case with HeLa cells. Tissue transplants are huge for our population these days, but we need to be careful with the credit and the legal side of it.